And also they are given filtered air using HEPA filter. So this machine controls the temperature and humidity, as well as it provides a HEPA filtered air to these cage systems where these mice are kept. So we have different types of mice. We have wild type mouse, which are the normal mice, where normal as in they don't have any kind of deletions or insertion or any kind of mutations. While we have knockout mice in which a gene of interest is deliberately deleted or mutated, and hence the gene of interest is not expressed throughout the lifetime of the mouse. While we also have conditional knockout mice in which the gene of interest is specifically deleted, specifically not expressed during the course of time, during a particular course of time in which we study the functional relevance of a particular gene. So we also use these animals to model human diseases, like suppose because of a mutation in a gene, there is any disease which happens in humans. So we delete that gene specifically in these mice models so that we can mimic the human disease in these mice models. For example, here we have a mice model of epilepsy.
Now, in addition to the reverse genetics approach, as I now explained, there we have mice models which are good examples of a forward genetic approach. In addition to reverse genetic approach, in which a gene is deliberately deleted, we have mice models which are examples of the forward genetic approach. In forward genetic approach, we have a mutation in the animal which can be seen through the phenotype, but we do not know what gene mutation or which gene is mutated that is leading to this phenotype. For that, various screenings are done to pinpoint the gene responsible for that phenotype. Once the gene responsible is known, we can have detailed understanding of the function of that gene by various other experiments. And if that phenotype of the mutation is related to some of the human disease, the mice can be used as the model for getting detailed understanding of that disease. For example, here we can see these two mice, both of them are born to the same mother and are of the same age, but you can appreciate the difference in their sizes. So this one is the obese mice and has very high blood glucose levels as compared to the other mice. So this obese mice can be used as the model for the diabetes and can be used to get detailed understanding of the diabetes, as well as to develop the drugs targeted against diabetes.
Hi everyone, so this is our quarantine facility, which also has the facility to edit the genome, as we have learned in our one of the lectures, the procedures to edit the genome. So here some people are working. Let's look into what they are doing. Hi Akansha.
Hi.
What you guys are doing?
Actually, here we are trying to create transgenic mice, which is a process of genome editing. Here we are using ICSI, which is intracytoplasmic sperm injection, where we are trying to insert our constructs in the testis of a 1-month-old mice. So here is Komal, and she is the technique.
So here, as Akansha told you, we are trying to do testicular transgenesis. This is mice which is undergoing anesthesia using isoflurane. So what I'm going to do is, this is the Hamilton syringe with which I am going to inject transgene into the testis of this mice. Let's see the procedure and
What this syringe is?
so this is special kind of syringe, Hamilton syringe, that does not damage the skin and can still enter into the skin and deliver our transgene directly into the germ cells of male. We target the germ cells present in the seminiferous tubule. So here is this. Akansha, can you please press the syringe when I keep it?
Okay, so can you zoom a bit more to see that dye? Okay.
So like this, we inject the transgene into the testis, followed by electroporation that involves giving 4 pulses of 60 volt each to both the testes, 4 reverse and 4 forward. So we'll keep the testes in between electrodes, and then we give pulses, 4 forward pulses and 4 reverse. Finally, we apply this providine iodine solution to the testes, so that any damage that has happened with syringe should not enter inside. And this mice is then kept for 30 days more so for recovery, and then finally after 35 days we set the mating of this mice with a female, and we expect the progeny to be transgenic.
So welcome all. We'll be looking into few model organisms which are already there in our department, and we'll talk to the respective people who are working on them. So can you come in? Just want to see. So Sriti, you work on mosquitoes as the model organism. So can you show us some, like how do you culture them?
So we have, these are the mosquitoes in these cages. We're going to tell you how we culture mosquitoes. So this is the very first stage. So these black dots, these are eggs. So we put these eggs in the water, and within 3 to 4 days they hatches into larvae. So these are the larvae. So it took around 8 to 9 days for larvae to emerge into the very next stage, that is pupa. So these these spherical, the round structures, the very small ones, these are the pupa.
What these black dots are?
They are eggs.
Okay, these are eggs.
And so these are around these. So larvae are in 3 stages. The first instar, which you are not able to see because they are very small.
Okay.
So these are around second instar larvae, and these are the third instar larvae. So after which they get converted to pupa, as I've already told you. Then these pupa, we keep them in the cages.
These are the cages.
So these are the cages. In these containers we have pupa.
Okay.
So within 2 to 3 days they emerges into mosquito. So these are the mosquitoes. So mosquito feed on sugar.
Okay.
So this is a
It's
sugar
called
feed.
flowers.
Yeah.
Okay.
So this is a sugar feeder. So we put 10% sucrose here. So
What
within-
is what is this?
this is basically here we store eggs.
Okay.
So after emerging into mosquitoes, within 3 to 4 days they emerges. We allow them to mate. So by 3-4 days they have already mated. So then we blood feed them using mouse. So these are the mice. So after blood feeding, then after 3 days of blood feeding, we collect eggs from them, and then the cycle continues.
So how this model organism is advantageous over the other, or what kind of questions can be answered using this model as mosquito?
So as you all know that mosquitoes are causing lots of diseases in humans like malaria and dengue. So our basic question is how olfaction plays a role in mosquito attraction to humans. Olfaction in the sense of smell. So we basically study how odors, which odors are attractive to mosquitoes, which are repulsive to mosquitoes, and our end goal is primarily to design some chemical methods or other methods which we can use to repel or kill mosquitoes, and not so that they cannot cause these diseases to us.
And do some, as our course is from functional genomics, so we are recording with respect to that. So is this you do whole genome gene expression analysis to identify like which orderant is causing enhancement or decrease in a particular gene, or such type of studies are also conducted with this model analysis?
Yeah, they are conducted, but we do not do them right now, but probably we'll do them in the future.
Yeah, in our lab we have 2 species. So this is a species which we got from Malarial Institute in Delhi, and this is another species, Liverpool. So they are sequenced. So their whole genome is sequenced. So such studies can.
Thank you. So this was Dr. Nitin Gupta's team from our department, and I thank you all. Thank you.
This is a room to maintain Drosophila stocks, and the room temperature is maintained at 18 degrees Celsius. So usually Drosophila is a cold-blooded animal, but the temperature for their viable conditions is 18 to 25 degrees Celsius. It will not be either below 18 or above 25. So this is one of the flasks of Drosophila flies that are climbing. So this is a plastic bottle. So below is the Drosophila food that we have put inside. The food consists of nothing but sugar, corn flour, and then yeast. So and of course, to make it as a semi-solid medium, so we add agar on this. So after solidification, we put the flies inside, and all the other activities like egg laying to the formation of larva and to the pupa, and the entire life cycle of this fly is in this bottle. So we maintain lot of genetic lines which are majorly we got from Bloomington Stock Center, Drosophila Stock Center in US. So and we maintain here. It depends on our need. This is a dissecting microscope. So what we have down here is the adult Drosophila flies. So these are the normal wild type flies, okay, just etherized using diethyl ether in a setup. This was just we made indigenous Drosophila etherizer. So it is nothing but a carton and then a perforated plastic vial, and over which a funnel was kept. And it is nothing but you put a diethyl ether 2 to 3 drops like this. Okay, 2 to 3 drops, and since it is volatile, so through perforated tube it will go inside, and now put your flies over down, so it will get etherized. So now these are anesthetized flies just kept here, and you can watch these flies under microscope very clearly. This is a female fly. Okay, so usually the female fly is characterized by bigger abdomens, and the size, especially the overall size, would be bigger when compared to a male fly, which is on the left hand side. So there are some body markers which we usually use to differentiate male and female, and this is a normal wild type fly which has a red eyed ommatidia, and there are many mutations which we used to generally developed and characterized. Has many genes which are mutant for body parts. Suppose if we have a mutant for a gene called white, the eye color would become white. This is one of the mutant fly. It has mutations especially in wing as well as in eye. The color of eye would be white, the antenna, and the ommatidial eye of the jot. So what we supposed to do is, before we'll transfer back and do fresh water. So this is this. For the next 45 days you can keep this. Yeah.
So do you also make mutants, or
usually in this lab we don't make mutants normally, because all these mutants we buy from Bloomington Stock Center. So they used to.
And-
Suppose if we want to generate any transgenic stocks, India we have a facility in CC camp in Bangalore, NCBS Bangalore. So there they will develop the
And what are
transgenic
the
lines.
techniques people use in Drosophila to edit the genome?
Yeah, main technique that we use is the P element mutagenesis. So there is a jumping gene that we call in biology called transposons. So these transposons are the majorly used in Drosophila generating transgenic lines. So what we have is we will genetically engineer these transposons. We insert our gene of interest. Then we trace it in a plasmid and give it to those facilities. What they used to do is in the embryo stage of Drosophila, we have a group of cells called pole cells. These are germline cells. So they will do a micro injection of these plasmids into it. So now this plasmid will integrate into Drosophila genome. So now the next generation of flies will carry this transgenic flies. So now we need to sort it out and then we will develop this. That's the major.
So thank you. So welcome. We are in the chicken laboratory where they use chicken as their model organisms, and we'll know from Tathagat how do they edit the genome or how do they insert foreign DNA into the chicken embryo. So I suppose this is the incubator where
So this is the 37-degree incubator where we keep our eggs so that they can progress with their stages. So when we incubate them, they're in the very early stages, stage 0, 1, and then they progress through the days. So chicken takes around 21 days to completely develop into a full-grown embryo. So what we do in our lab is, we have two labs, one working in brain and the other one working on bone. So we use viral DNA and we electroporate the viral DNA into the chicken tissue. And because it's viral DNA, the viral DNA integrates itself into the chicken genome, and then we get the necessary phenotype of each of the gene that we have studied or we want to study.
Can you open this actually?
This is the incubator. You have different students having different plates, and as you can see there is a window. If I show one of the plates. So what we do is we open a small window and then we manipulate the embryos inside, and then we seal it up again with a sellotape, and then we again keep it back for the rest of the days according to experiments. Like if we need it in an adult embryo, then we'll keep it for maybe 10-12 days. If you need it early, then we can harvest it in within 3 days. So that's it. So now we can go to the electroporation. There's a student who is already performing electroporation where we introduce the DNA with help of voltages. So DNA being negatively charged, so what we do is we inject the DNA into a pouch, maybe in the limb pouch or in the brain pouch, and then we use electrodes. It's almost like giving a shock, and the DNA moves inside the cell, and then we have the genome ready for being manipulation with the viral DNA. So this is our electroporation chamber, and this is Baba, MTech student of our lab. Now what he is doing is he is using the electrodes to put in his.
You want to demonstrate? Yes. HH12 आपको कुछ करना है HH12?
So this is the entire unit. This is the micro injector through which we introduce. You show. So this is the micro injector where we use it to inject the DNA. So you have this magnetic base with which we stick it onto the base, metal base. You have the capillary tube in which we have our DNA solution, and we use pneumatic pressure pump through which we force in the DNA into the embryo. So I will just use one of the eggs. What we do is, at first we lower the embryos. That is, we take out some amount of albumin from the egg so that the yolk settles down, and then what we can do is we can cut one small window onto the top. And you can see the yolk. Now this is very early stage embryo, and you won't be able to see much of the embryo, but there is an embryo which is developing. It's very early stage 10. So it's like almost day one of incubation. So it's just a speck of line. What we do is we manipulate everything that we want to do through the microscope, and then we seal it back up with the sellotape. So I'll just show what we do. The first thing that we do is we'll set it up, look into the microscope, and we'll aim our microinjector onto the embryo. So this gives us maneuverability top and bottom, left and right, and with each press there's a pressure going into the pneumatic pump which forces DNA into the tissue. We have a dye, as you had seen earlier, that it's a blue dye which we use so that we can see if the DNA is getting into the embryo properly or not. And then when it's finally done, we have two electrodes here, and we direct the DNA towards the positive end because DNA is negatively charged, and from the other end we place it just at the bottom of the yolk. So we place one electrode at the bottom of the egg and the other one would be on top, and then we have another switch here which we operate with our leg, which would have this electroporator unit which is now set at 10 volts on. As you can see, it's the on time is 50 microseconds and the off time is 950 microseconds. So it's the pulse rate of the voltage pulse that we give, and it goes on for a total of 5 cycles, that is 5 seconds. So with one press, it's the second count. So it's one full pulse of 5 seconds which goes in, and we use it for putting the DNA into the cell. Once it gets inside the cell, we seal it back up. And we keep it for incubation. Now obviously it takes around 8 hours for the gene. Say we use a fluorescent gene, a GFP or mCherry. So it takes around 8-10 hours for it to be shown. So what I can do is I can show you some images which finally will find out the limb. Say you have electroporated in day 2. I'll just show a limb which is having green fluorescent protein. So you'll have the entire limb completely green. So as you can see, we are putting in green fluorescent protein into developing. As you had seen earlier, it's only a streak at that point of time. But if you let it stay on for a longer time, like this is a day 8 embryo. So this was the limb in which I had incorporated the RCAS GFP. It's a viral vector with green fluorescent protein along with it. So because it's a viral vector and it's a virus essentially, so it spreads along the limb. And now the entire limb which I had electroporated is fluorescent green label. As you can see, this is the green limb, as you can see, and this one is the other one which doesn't have any such green fluorescent protein. So instead of fluorescent protein as I've used here, I can use different genes to study different aspects of bone development, and there are several other, like I use different genes to have different kind of phenotype and study how they are. So, and the similar thing is used for brain development.
Hi Nilesh.
Hello.
Yeah. So with respect to functional genomics, as in this model fish, how do you maintain this fish model, and how do you edit the genome of this basically?
For editing genomes, there are various technologies like most recent one is CRISPR-Cas.
That can be used in liver-fish.
It can be used. It has already been optimized for fish system. There are killing methods. Then there are toll-to transposon-based methods available. We have CRISPR.
And why this one is specifically red? Is it like the natural color of the fish?
No, that's not natural color. This is alpha-actin A RFP.
Oh, this is the RFP model. So red fluorescent protein. It is expressed.
It is.
And this one.
This is a Casper model. Basically, the skin is transparent in this model. So all the internal organs are visible without doing anything. In a live organism, you can see liver and other.
Can you show us one fish? Some this transparency of the body. Is that
Sure.
we-
I can. So, give me a moment. So this individual is a female, and if you look at from sideways, the bulk white portion towards the tail are the eggs, and the front portion, which is a bit yellowish, that contains liver and nearby organs, pancreas. You see a red line going from front to the back. That red line is basically the fish kidney.
So hope you enjoyed the lab trip. So the idea was to give you an kind of overview as to what are the facilities people use for genomic applications. And with this session, we are concluding the 10-hour course called Functional Genomics. I hope you have enjoyed the course, and if you have any queries or doubts, you are most welcome to write to us. We'll try to address, but we will also be taking up one last hangout session, so we'll be able to interact more.